Developed in the late 1980s, the polymerase chain reaction, or PCR, was first used to characterize the mutations associated with sickle cell anemia through the work of Randy Psyche and Karen Mullis. PCR is an important tool for molecular biologists today because it allows us to not only amplify but also detect short fragments of DNA in vitro. It has many roles in molecular diagnostics, cloning, as well as it's allowed us a better understanding of the molecular biology behind disease. This talk will mainly focus on four main topics, so the components and the steps of the PCR reaction, results visualization using gel electrophoresis, as well as the application of PCR in the real world. Welcome to my TED Talk. It's an introduction to PCR. On the screen, we see a variety of components that are required for a PCR reaction, many of which you'll actually be familiar with because they're required for DNA replication in vitro. In a buffered solution, we still need primers, deoxyribonucleotides, a template to copy, and some cofactors, but as well as a special kind of DNA polymerase, a heat-stable DNA polymerase like Taq polymerase, which was isolated from a thermophilic bacterium like Thermicus aquaticus. Higher temperatures increase the yield of the reaction, as non-specific products, such as different like primer duplexes, were less likely to occur. There's actually three main steps associated with PCR, which can be carried out in volumes as small as 30 microliters per reaction. You can have the denaturation of the template DNA, primer annealing, and then eventually extension. So what you need is a thermal cycler, so the reaction is subjected to really high temperatures, approximately 95 degrees Celsius, to separate the parental strands by breaking the hydrogen bonds between them. After lowering temperatures to about 55 degrees Celsius, primers can then anneal and bind to provide a free 3' hydroxyl group to facilitate Taq polymerase extension. This process may be repeated for about 25 to 40 cycles, such that like one single DNA molecule could be amplified to more than 80 nanograms of the product. It's important to consider that the annealing temperature is often subjected to a lot of optimization so that we can minimize the presence of different contaminant species. So after PCR, the amplification of your gene of interest can be seen by separating out your PCR products on an agarose gel. DNA, which has a negatively charged phosphate backbone, will move from the cathode to the positively charged anode. Ethidium bromide or cyber green can intercalate between the bases to allow for fluorescence base detection. So a lot of your larger fragments can be seen at the top of the gel, whereas a lot of your smaller fragments will be closer to the bottom of the gel. So the polymerase chain reaction is important for cloning and molecular diagnostics. PCR can amplify a region of interest, which can be inserted into a vector with different promoters for bacterial transformation protocols. It also serves as an important role in pathogen discovery. So for example, in a review by Gelder, PCR can serve as a tool to diagnose CMV infection, where other laboratory techniques had difficulty before. Similarly, diagnosis of cystic fibrosis has been shown to improve when we're able to sequence the entire gene itself, as well as do real-time PCR analysis. So what we're able to really deduce from this is that the possibilities are endless.